For the past couple of chapters, we've been discussing how information, inherited information, is passed generation to generation, starting with Mendel, going to Morgan, going to Hershey Chase, Watson and Crick. Now for this chapter, chapter 15, we're going to be discussing how that information is read. Okay. The DNA is the stored blueprint, but the blueprint itself does not make the organism. Okay. The blueprint is instructions on how to make proteins. Proteins are what will become the cytoskeleton, what will become the enzymes that make the lipids, that will become the membranes, that make the enzymes, that will make the pigments, that will give you the skin color, the eye color. The reading of the blueprint to make the protein, or more correctly, the polypeptide, this is what is referred to as gene expression. It's the reading of the DNA strand and ultimately ending at a new polypeptide. Now, why do I keep saying the polypeptide? If you think back to an earlier chapter when we were talking biomolecules, primary, secondary, tertiary structure is the folding of the polypeptide. Quaternary structure is either the final polypeptide shape or it's the combination of multiple peptides to give you a complete protein. One gene, one polypeptide. It all started back early 1900s. This idea that the information in the DNA, still before we knew exactly what, that the information in the chromosome, before we knew DNA, before we knew all the proteins that are involved there, we were still able to determine how this worked. A British scientist, physician, by the name of Archibald Garrod came up with this idea of studying cells that were mutated to see if they could or could not produce certain proteins. This idea was later carried on by the research lab of George Beadle and Edward Tatum. You'll hear them referred to as Beadle and Tatum. They took this idea that, Garrod had, gene, protein, and they wanted to look at it and see if how can we go in an experimental fashion, not only prove this, but study the mechanics, how this works. Beadle and Tatum took mold, just plain old bread mold, and they bombarded it with X-rays. X-rays cause DNA damage. Some of that damage will be repaired, but not all of it. So they were intentionally trying to create mutants. They took the mold that was irradiated and they put it on agar that was lacking certain amino acids, lacking certain components. Non-mutated strains would be able to grow, would be able to compensate by synthesizing the cell's own amino acids and whatnot. They were able to show that certain mutations These mutants could not grow because they lost the ability to produce certain amino acids. A mutated gene was responsible for an enzyme that was part of the pathway involved in that amino acid production. Serb and Horowitz took Beadle Tatum's work a step further, and they started examining specific mutants, specific mutations. And they studied an arginine-deficient mutant, meaning that this mutant was incapable of synthesizing its own arginine. They found that the mutations for the different strains occurred in different places on the chromosome. Even though the mutations occurred in different places, in different genes, the mutant was still incapable of producing arginine. From this, they were able to correctly deduce that the production of arginine is not a one and done, that there were multiple steps involved, what we now think of as a metabolic pathway, that by going through and mutating different genes, they could interrupt at different steps. Their results supported the one gene, one enzyme hypothesis. Okay, one gene, one protein, one gene, one polypeptide.